everyone, and welcome to today's PHO Rounds presentation on scales, tails, and salmonella, a comparative analysis of human and reptile isolates. My name is Christine Navarro, public health physician, and I have the pleasure of moderating today's session. It is now my pleasure to introduce the speakers for today's presentation, Dr. Katherine Paphitis and Dr. Alexandra Reid. Dr. Katherine Paphitis is the enteric and zoonotic specialist at Public Health Ontario. Her primary research interests are One Health and the epidemiology of enteric and zoonotic diseases, including salmonellosis. Dr. Alexandra Reid is a lead veterinarian in the Ontario Ministry of Agriculture and Food. Her responsibilities include regulatory response and emergency management, exotic, aquatic, and alternative species, and wildlife domestic animal interface issues. And now I'll pass it on to Dr. Reid.
Thank you. So we're here today to talk about scales, tails, and salmonella, a comparative analysis of human and reptile isolates that Katherine and I have been working on for the last year. Next slide, please. And so just briefly, this presentation was created by its authors, and we're going to put it on the Public Health Ontario website for public use. It's outlined in website terms of service. We're not the owner, and just there's no liability resulting from any such application or use. Next. And then for learning objectives, we're just going to ask that you learn to list common salmonella serotypes, describe the risk of salmonella transmission risks associated with reptile ownership, identify the association between salmonella reptiles and feed or prey to support outbreak investigations, and identify practices that can reduce the risk of infection from reptile contact.
Okay, so thank you for the introduction, Christine. Today we're speaking about Salmonella, which is a zoonotic disease, meaning that it's naturally transmissible from animals to people. So for those who are unfamiliar, we wanted to note that there are many different types of zoonoses, and people may be exposed to these in various ways, including through indirect or direct contact with animals or through ingestion of contaminated food or water. Risk factors for infection differ, but for Salmonella, we know that some population subgroups, including the very old and the very young, may be at increased risk of infection and of severe illness if infected. So to give you some background on the epidemiology of Salmonella in Ontario, after Campylobacter, Salmonella is the second most commonly reported bacterial cause of human enteric illness. And although there are over 2,000 different serotypes of Salmonella, only about 100 of these typically cause most human illness. So for example, in 2023 in Ontario, there were almost 2,400 cases of Salmonella reported, with almost half of these due to just 3 serotypes. So collectively, 3 serotypes accounted for almost 50% of our cases, and those were Salmonella enteritidis, Salmonella typhimurium, and Salmonella branderup. So although there are numerous different serotypes of Salmonella, relatively few of these account for most human cases. Following exposure to Salmonella, the incubation period or time from exposure to symptom onset is variable, but generally in the range of 6 hours on the shorter side to up to 7 days on the longer side. So public health investigators typically focus on the 1-week period prior to symptom onset when attempting to identify a potentially causative exposure. Lastly, non-typhoidal Salmonella has a wide reservoir range, including many animals that we may keep as pets, such as reptiles, cats, and dogs, just to name a few. So just to give you some background context for today's session.
So when we talk about reptiles, it's really sort of important to know what you're actually dealing with. It's not a very homogeneous group of animals. And so typically when we talk about reptiles, there's sort of some common groupings. So one of them are the squamates. And so the ones we think of are mostly what we call lepidosaurs. They're snakes and lizards, but this branch of the reptile family also includes tuataras, which are a very rare and very localized sort of family of very primitive reptiles that are only found in sort of the New Zealand area. So typically when we think of pet snakes that are out there, we think of ball pythons, carpet pythons are very common. Boas, corner rat snakes, and king snakes are some of the more common ones you might encounter. And then lizards, again, there's a lot of diversity in lizards that are kept in terms of size and diet. They can, leopard geckos, for example, are very common. Crested geckos have exploded in popularity. They were actually not known in the world as a species until 1990, and they're probably one of the most common lizards kept today. Bearded dragons, monitors, and tegus are sort of larger predatory lizards that some people keep. And then colonia are the turtles and tortoises. So typically we think of turtles as animals that spend some time in the water, and tortoises as strictly land-based. There's also the more anatomic sort of division between what we call side neck turtles and then the cryptodeers, the ones that actually pull their head into their shell. But common turtles that people might keep include red-eared sliders. They were the ninja turtles of the 1980s, if you were in my age group. Alligator snapping turtles are fairly common. They get very, very large. False map turtles. And in tortoises, there's leopards, there's hermins are very common. They're a smaller species. Russian or Greek tortoises get a little bit larger. And then the sulcata or spur-thighed tortoise is the third largest tortoise species in the world, but they're very commonly bred and sold. And then crocodilians are not illegal in Ontario. There's only legislation at the municipality level. So there are quite a few here, less common than others because of the space and the dietary requirements. Spectacled caimans are probably the most common species that you'll see. Dwarf caimans are popular as far as they go because they stay under 4 feet total body length. And then American alligators are actually very common in the pet trade and do reach very large adult sizes but are fairly docile. Next slide, please. So things that are not reptiles are amphibians, fish, birds, even though some of the bird people say that they're technically dinosaurs and dinosaurs and reptiles are somewhat related. Really, all that sort of unites reptiles as a group of animals is they have thick, scaly integuments that must be shed in some way, and they're dependent on external heat for their metabolic processes, but they're not necessarily at the same temperature as the environment. So it's important to realize that a lot of reptiles should feel quite warm to the touch. They're not cold, they're not slimy. Um, they're, you know, very sort of interactive with their environment. Next slide, please. So salmonella can be a normal component of reptile gastrointestinal flora. So not every reptile always has salmonella, but it can be sort of sporadically detected in reptiles. It can also be a very significant pathogen for them, especially when they're already stressed or sick or have some other underlying process. So the salmonella that makes them sick can either come from within, from their own gastrointestinal tract, or it can come from external sources like their food or their environment. So most normal healthy reptiles will never show signs of gastrointestinal colonization, right? They just sort of trek along and you would never know it was there. But if there's suboptimal husbandry and stresses, like inappropriate food or excessive handling, it can really increase the likelihood of shedding. And then stress can also permit extraintestinal colonization. For example, in snakes that are under stress, we know this shifts their oral flora towards gram-negative and promotes colonization by salmonella in their mouth, which can lead to infections. We do not recommend trying to eliminate salmonella either from adult reptiles or their eggs. It's very antibiotic therapy doesn't really seem to resolve the carrier state in any way and just promotes antimicrobial resistance. Reptile eggs are not like bird eggs. They're very leathery and soft and permeable. So anything that you apply to the surface of the egg, like an egg dip that we might do with like a hard shelled chicken egg, can actually kill the developing fetus. Next slide. So common signs of suboptimal husbandry when you're looking at a reptile and sort of an outbreak and saying, is this potentially a sick reptile? Um, Things like lethargy. A normal reptile should look at you when you walk in a room. It should be moving. It should show interest in what's going on around it. If they're just kind of sitting there and they're not really lifting their head up, they're not interested, that's a bad sign. Often they won't eat. And so the owner might say, yeah, it's not been eating very well the last little while. If you see a lot of sort of retained skin patches, like all of us in the winter, sometimes reptiles need a little extra help when the humidity drops. But when you can see layer upon layer upon layer of retained skin, there's a problem there. If you see loss or discoloration of the toes or the tail tips, that's quite common in sick reptiles, especially with that retained shed. You might see patchy erythema, particularly along the belly, along the bottom of the throat. Snakes, often it's the full belly or right around the cloaca. Um, and sometimes you can actually appreciate petechial hemorrhage on the reptiles as well. Sudden death or sepsis is the most common presentation of salmonella infection, although clinical presentations of salmonella sort of overlap with a lot of other things. So most bacterial pathogens that cause sepsis could kind of look the same in reptiles. It's very common for pet reptiles to have concurrent nutritional deficiencies, even with a very good owner. A lot of them have very complex dietary needs that are very difficult to simplify down into a household. If you live in a small town, you may not have the same access to like stores and diversity of diet that you could get in a larger center like Toronto or Ottawa. Um, so even with good intentions, often reptiles are clinically deficient in one or more vitamins and minerals. Snakes very commonly show severe stomatitis and osteomyelitis as a primary form of salmonella infection. Um, Snakes with oral infections, their bone is right under their gum, and so it's a short hop for oral colonization to turn into osteomyelitis, and from there it can access the bloodstream. Other species, when they show signs of sepsis, like those petechiae and lethargy, they may also show pneumonia. So you might see reptiles with nasal discharge, for example, or they blow bubbles from their mouths. You might see abscesses growing along the body. Sometimes they rupture and they have sort of thick caseating pus. Again, they're lethargic, they're not eating. Sometimes you see cloacitis or pericloacitis. Reptiles have a lot of glands right around their cloaca, so you can see infections in there as well as their external reproductive organs. And then osteomyelitis. So, very commonly in snakes, often you see spinal deformities. It looks like their back is broken. Um, Lizards, too, you can kind of see bumpiness of the spine or the tail or the limbs, often swelling of the sort of foot area. Next slide, please. So in Ontario, we get reports of salmonella that's isolated from reptiles. So private veterinarians can submit clinical samples from reptile and amphibian patients in Ontario to veterinary diagnostic laboratories as part of animal health exams. So typically, these specimens would only be submitted from sick or deceased animals. Again, the value of trying to test an animal for a carrier state is low. So usually there's a reason that a swab is being sent in. um, Salmonella positives get reported to us. So depending on the lab, we might just get a report that, it was PCR positive for salmonella. If it was done as a culture, we do get all isolates serotyped and reported to us as well. So this lets us assess the common serotypes reported from reptiles and the species associated with them. Sometimes pooled feces from live animals can be tested with enrichment. We tend not to get excited about these because shedding is intermittent, and really, if you need enrichment to find salmonella, it only says it's the normal carrier state. So it doesn't really say that there's a problem with that reptile. Many of the samples we get reported to us, for example, come from internal organs on postmortem or from wounds, abscesses, that kind of thing, and that's more likely to yield a pathological salmonella. Next slide, please. And so turtles have been typically overrepresented in salmonella outbreaks. In the United States, the U.S. Food and Drug Administration banned the sale of turtles with a shell length of under 4 inches to the public in 1975 because turtle-associated salmonella infections were so common. It's thought that the really small turtles, they're not only easier for small children to handle, but they fit in their mouths. Um, so national outbreaks linked to small turtles sold illegally in the United States continue to occur. They're allowed to sell them for scientific supply. So, with websites, people can order these things for themselves. And so, there's real enforcement challenges with trying to sort of enact this type of legislation. Ownership restrictions in Canada they're decided at the provincial level. In some provinces in Ontario, we have no provincial level animal restrictions. So all ownership restrictions are at the municipal level. For example, in Guelph, it's illegal for me to buy a turtle, but I can go to Kitchener and I can buy turtles no problem from the pet store. And they generally have a selection of species available at some of the bigger pet stores. So there's enormous diversity in what reptiles may eat, and some of these species may eat most of them. Some of them may have very specific prey requirements. So for feed or insects available in Ontario, you can get two species of cricket, the house cricket and the banded cricket. You can get discoid roaches. Some people may also have dubia roaches, which are technically illegal, but they look very similar to discoid, and they were both here probably illegally before CFIA approved discoids. There's mealworms, which are these little guys in the top right here. Superworms look the same, they're just bigger. There's hornworms, which are the sort of green caterpillars that many of you might pull off your tomato plants, waxworms, which are a moth larvae, black soldier fly larvae. There's increasing interest in them because, unlike other crickets or other insects, they actually have a positive calcium to phosphorus ratio, which is very important in reptile diets. And then there's two species of fruit flies, commonly raised as sort of wingless fruit flies, Hydei, which are slightly bigger, and then melanogaster. So these might be raised on scraps like roaches and black soldier fly larvae. It can be raised on, waste from your kitchen. The black soldier fly companies that are out there and raise large numbers often use things like restaurant waste, or they might have really specific diets like crickets and fruit flies can actually be maintained on like lab animal powders that are reconstituted and fed to them. Insects themselves can be natural reservoirs of salmonella, or it's very easy to contaminate these colonies, right? You can't add a lot of antibacterial agents, for example, to fruit fly larvae, or you'll kill your fruit fly. So these are sort of high nutrient systems that can become contaminated, and at a factory level, you can potentially um, contaminate the entire system. So there's the potential for insects to contaminate a lot of reptiles. And next slide, please. And this is just a picture from one of my bulk cricket orders showing how sort of dirty these systems can be when you get them. So you can see in the middle, there's this egg crate that's covered in adult house crickets. And then on the egg crate, once the crickets have been knocked off, you can see it's just speckled with feces, and the same, they've been in the box on the left. That's sort of how they arrive in these cardboard boxes full of egg crate. It's once again just coated with feces. And I think a lot of people might not realize that insect feces pose a risk to them, and you can see I'm wearing gloves, but some people, might not um, have the same sort of level of concern when they go to PetSmart and buy a bag that just has a little piece of egg carton, and they don't realize that potentially that's something they can make themselves sick with if they're not careful. Next slide, please. And then for feeder vertebrates, it's really important when you're looking at some of these production systems. Remember, it's not just reptiles that eat them. We have a lot of exotic cat owners in Ontario, for example, who will buy these for either enrichment or the basis of their diet. Ferrets, again, as obligate carnivores, they're basically mammalian snakes. So a lot of them will be fed, mice and rats. And people who um, have raptors for various reasons, either rehabilitation or if they actually like keep and breed things like peregrine falcons, they'll be feeding them these in the off hunting seasons. So common rodent species used as feeder vertebrates, there's quite a few. So in Ontario, it's often the house mouse, which is the basis for the laboratory mouse. The Norway rat, right, the brown rat. We don't see a lot of breeding of black rats, but they are out there once in a while. And then very popular right now are the African soft-furred rat, which is also called the multi-mammate mouse. It's an African rodent that's kind of not quite a rat, not quite a mouse, but kind of looks like a rat. These are generally, as a group, raised in very high density colonies, often in sheds with additional ventilation and sort of these vertical homemade racks. They're fed a mixture of commercial pellets, like you can buy lab animal chow, it's called, with or without dog kibble for added protein. Some people might just feed bird seed, which isn't ideal. Often they have these sort of DIY gravity fed water systems that go to all of the racks. The biosecurity is really low in these. They're often really high in ammonia. They've often got wild rodents, wild insects accessing them. Escapes are common, and they might just sort of throw them back in the tub that they think they came from. And a lot of these come from the pet trade, so they're not coming from like high health lab animal stock. They're coming from whatever someone thinks looks healthy when they decide to start breeding rodents. So generally very low biosecurity, and probably a lot of these are colonized with salmonella. There's actually a best management practices document for feeder rodent production and distribution that you might find helpful if you're ever investigating one of these sort of larger rodent colonies. Next slide, please. There's even more feeder vertebrates out there too. So chicks are very common. They might be culls from the layer industry, like male chicks that don't end up becoming breeders, or they might be raised specifically for reptile food. Most of them are probably chicken chicks. There is a fair number of quail chicks out there because they're smaller. Some people do sell ducks that they're again either getting from like one of the duck commercial operations here, or they're raising them specifically for reptiles. There are a lot of larger reptiles out there that might consume guinea pigs, rabbits, and or small pigs. These pigs sometimes are stillborns too, so they may have underlying health issues at the time that they're frozen back for these reptiles. And then there's also live feeder fish. So in the pet trade, they're generally cyprinids. So things like bait fish that you can buy to go fishing. Some people feed small goldfish, what are called rosy reds or fathead minnows. But sometimes there's these hybrid live bear species like they look like mollies or guppies. Long term, a diet of these baitfish species can lead to severe nutritional deficiencies, particularly for thiamine. So sometimes neurological disease is very common in reptiles that are fed these animals exclusively. Next slide, please. And then most of these things are killed either with carbon dioxide, which is not AVMA recommended because it is adversive to rodents, or they might simply be frozen alive. And they're often frozen as a whole animal. They're not processed in any way. Feeding live prey can actually increase oral trauma from being attacked, right? They don't want to be eaten, they'll be biting, especially for a snake that pretty much can only handle an animal by putting it in its mouth. And sometimes, if they're not hungry, especially like a hungry rat, can turn around and attack a reptile and cause significant damage. Some people also have very poor freezing practices, like anything else. These animals are warm when they're first killed. If you pack them too closely together, really stuff your Ziploc bag full of rat, it takes a lot longer for them to freeze than say someone who spreads them out on a tray, increases their surface area, lets them cool quickly, and then packs them for freezing. And it's just important to remind people that freezing and drying do not reliably inactivate or kill salmonella. There is a perception out there that reptiles cannot get salmonella in the pet trade, or that freezing magically removes pathogens, and it does not. So there's still a fairly high risk practice. Next slide, please. And then there's all these new diet formulations out there that I think are important to know about. So powder to gel diets are a mainstay for some species. So those crested geckos that I highlighted, you buy these powders and you mix them with water to kind of a milkshake-like consistency. That's the pile of goop in the photo. So these animals would then lap them up. Some of them actually set to more of a firm jello consistency for omnivores. Is increasing in popularity as a supplement for other species, either to diversify diets or for convenience, right? If you live in a small town, you can order this off Amazon and then you don't have to worry about going to a pet store and getting crickets and then housing crickets, and do the crickets get out in your house? These things are made from a blend of protein sources, like often it's egg powder, powdered insect meal, powdered fruit, other carbohydrates, vitamins, and mineral supplements, and then it's mixed with water. And so there's also increasing interest in what used to be called sausages. They're basically an extruded carnivore diet that might incorporate fish meat, frog meat, poultry, or rodents with vitamin and mineral supplementation, and then it's set into a thick gelatin shaped and cut. So these will be stored fresh and frozen. The problem with both these kinds of diets is they're not really produced with the same regulatory oversight, that say a baby food would be made with, right? As you're making these microbial media from ingredients that might be contaminated already, and then there's no additional food safety handling. If you look in small print on the package, it'll typically say dispose of uneaten food within 24 hours. But just having sort of an open container of this food type, it's very easy for your reptile to contaminate with feces. Do you wash this out in your sink? There's important food handling, I think, considerations that don't always get highlighted when people are feeding these. Next slide, please. And then there's also home prepared diets. So rats are expensive, chicks can be expensive, rabbits are fairly expensive because they're usually also produced as either pets or for human food. So they might take an expensive item like a feeder rat. A large rat's maybe $6 right now, and they'll cut it with a grocery store product like organ meat, eggs, which we know can be contaminated with salmonella, might be egg product even, drumsticks, ground meat, or processed carcasses. So this is a picture of some beef heart that I was cutting up to give to my turtles for enrichment. Many herbivorous animals are fed green produce from the grocery store. Some people will feed things that have expired to their pets because they think it's somehow safe for them, even if it's spoiled or moldy or something that says it's probably not something you should be consuming. Some people also use like baby purees. There's this belief that you can take um, baby puree and mix it into other things, and it becomes like the gastrointestinal contents of, a rodent or a chick, which isn't true. And there's also recipes out there. So there's this so-called San Diego Zoo recipe that some people use, even though the San Diego Zoo says they have nothing to do with it. But for a very large carnivorous lizard, like some of those monitors and tegus, they take ground turkey, baby food, and a human vitamin and a and they mix that together and feed it to their reptiles as is. And if they're buying big chub packs of ground turkey, maybe they're making quite a bit in one go and feeding it over a week. So some of these can be really highly contaminated in the first place. We certainly know commercial grocery store meat is not safe to eat raw. Um, and they may not be handling these things well. I mean, even bagged salad mix, how many food poisoning outbreaks can be associated with that? Next slide, please. So moving from reptiles to people, this is this Katherine slide.
Yeah,
I will
thanks.
move.
Thank you, Alex. So as Alex mentioned, it's not abnormal for Salmonella to be present in a reptile's gastrointestinal flora. And if present, the bacteria can be shed into the surrounding environment. So people might be exposed to Salmonella either through direct contact with their reptile or, as Alex has nicely teed up, through indirect contact, including through handling the reptile's environment, the packaging from their food, or the food directly itself. So it's also important to be aware that the food fed to a reptile might also be a source of Salmonella, and that a person can similarly be exposed to the bacteria through cross-contamination or poor hand hygiene, such as using the same cutting board or utensils for the reptile food preparation as well as for human food preparation. And it's not uncommon, depending on the type of reptile, for pet owners to allow their pets, particularly bearded dragons, I know there's a lot of bearded dragon folk in the audience, to allow them to roam outside of their enclosure. So if they're allowing them to roam on kitchen counters or bathroom counters, for example, that is a higher risk of contamination for the humans. So ideally, making sure if you do allow your reptiles to free roam, that you're making sure it's on environmental surfaces that are easily able to be cleaned and disinfected. So for those involved in Salmonella case investigation and case interview, if a client reports they did have reptile or amphibian contact during the exposure period, we'd recommend gathering as much information as possible about the type of reptile, its diet, and how they typically handle both the reptile as well as the enclosure and the feed. This can be very helpful in determining whether the reptile might have been the cause of exposure, which we'll get into in a second. And for example, when I was younger, and I promise pre-learning about public health, I had a pet frog that I was told by the pet store needed to eat frozen pinky or fuzzy mice, which Alex has since told me was not the right diet for my pet. But at the time, having no food safety or zoonotic disease background, I used to thaw my mice on a kitchen plate that I also ate from, not at the same time, in my microwave. And periodically they would explode, and I would just give my microwave a wipe down and go on with my day. And in retrospect, I was likely regularly cross-contaminating my own food with mouse bits and pieces and was blissfully unaware that I was doing so. And I'm sure I'm not the only one that has made similar, or I hope I'm not the only one that's made similar food handling mishaps just from a lack of awareness about the fact that these can be a source of contamination. So case interviews also provide an opportunity to provide education to clients and to find out if there are other susceptible individuals in the household who might also be at risk of exposure. So for case or outbreak investigations where reptiles are a suspected source, we have found at PHO that testing of environmental swabs may be helpful. So if this is something as a public health unit that you're considering, please contact PHO's laboratory before submitting any specimens, and our lab folks can provide you with collection and submission instructions. In previous investigations, as Alex has alluded to, we found that fecal specimens are not very helpful, as we know that Salmonella shedding can be intermittent. So if you do collect a fecal specimen, it's usually weeks after someone's exposure actually happened, and a negative specimen doesn't rule out the reptile as the source of human illness just because of that intermittent shedding consideration. Environmental swabs, however, collected from high-touch surfaces within the reptile enclosure, such as food dishes or preferred seating areas, have been found to be very helpful in our previous investigations. So environmental swabs are tested by bacterial culture at PHO's lab, and if Salmonella is identified, serotyping and whole genome sequencing are performed. Typically, isolates are considered related within 0 to 10 whole genome multilocus sequence typing alleles. And for zoonotic outbreaks, however, we might use a wider allele range just to reflect the higher genetic diversity that's associated with such outbreaks. So as reptiles have become more popular as pets, there's data out there showing that particularly bearded dragons and lizards are becoming more popular. The number of reptile-associated outbreaks has also increased. So on the slide here are some of the recent national outbreaks involving Salmonella in reptiles, and for which the Public Health Agency of Canada has issued a public health notice. These have included outbreaks of various serotypes linked to lizards and snakes. And you'll note that those linked to snakes also note feeder rodents as a potential source, as it's often difficult, as Alex mentioned, to differentiate whether the source of human illness was contact with the snake or with the feeder rodents, recognizing that both are a reservoir for Salmonella. I've also included a recent outbreak investigation in the United States linked to small turtles, reflecting the challenges in enforcing a ban on these as pets, as Alex mentioned earlier. So, what is all of this leading up to? Well, as you can probably tell, Alex and I both have a particular interest in reptile associated salmonellosis. So, we decided to do an analysis of our human and reptile data, so the human case data reported to PHO and the veterinary isolates reported to OMAFA, to see if we could identify any associations between specific reptile types and serotypes responsible for human illness, or between their diet and the serotype isolated from the reptiles. So, to do this, we took all of the cases of human salmonellosis reported in Ontario from 2015 to 2022, and we had the implicated Salmonella serotype, date of symptom onset, and whether or not each case reported travel and or contact with reptiles or amphibians within that 7-day period prior to symptom onset. And we then compared the serotypes and reptile types reported by our sporadic, so local acquisition cases with no travel history and those not linked to a known outbreak, to reptile isolates reported to OMAFRA during the same period. If anyone is interested in reading this work in full, we don't have the time to do it full justice today, but it has been accepted for publication in EID, which we're very excited about, and is out next month, I believe. So we'll give you some of the main highlights. So to give you an idea of the data we were working with, we started with over 18,000 human cases of Salmonella that were reported in Ontario during that time period. Almost 70% of these did have a yes or no response about reptile contact during the exposure period. So for the remainder, unfortunately, this question was either not asked or the field was left blank. Of the almost 13,000 cases that did have a yes or no response, most were considered sporadic, meaning there was no epidemiological or whole genome sequencing linked to other cases, and 7% were linked to almost 100 outbreaks, 2 of which were reptile associated. So once we dropped the folk who reported travel, which we did on the basis that the types of reptiles and reptile interactions that someone might have while on vacation, for example, likely differ from those with reptiles they keep as pets, that left us with 513 human cases of Salmonella who did not travel during the exposure window, but did report having contact with reptiles and amphibians. Before focusing on those 513 cases, however, we first looked to see after removing the travelers, but before removing the cases with no reptile contact, if when we only looked at those sporadic cases, so those that weren't linked to a known outbreak associated with chicken or some other type of source, if there was a difference in the age distribution between our sporadic cases who did report reptile or amphibian contact and those who did not. And interestingly, we did find a difference, which you can see here. So the cases who reported reptile or amphibian contact are shown in green, those with no contact are in blue. And as you can see, the folks with reptile contact skew more to the left, and it does appear that the Salmonella cases with reptile or amphibian contact were generally younger compared to those with no contact. So to quantify it for you a little bit, just under 10% of those who reported reptile or amphibian contact were under 1, compared to just about 4% of those with no reptile or amphibian exposure. So for the remainder of our analyses, we focus specifically on those 513 cases with no travel, but who did have reptile or amphibian contact. So when we looked at the reptile types cases reported having contact with, lizards, interestingly, kind of paralleling what we saw in our poll to you today, lizards including bearded dragons, geckos, chameleons, iguanas, and other lizards were most commonly reported, followed by snakes, then turtles and tortoises, and amphibians last. Interestingly, 12% of our cases reported having contact with more than one type of reptile, and 1 person reported contact with a crocodile. When we looked at the serotypes that we identified from our human cases, there were 94 unique serotypes, almost half of which were only reported from a single individual. There are 6 different Salmonella subgroups. However, most human illness is actually caused by Salmonella enterica subspecies enterica, and this was reflected in our data where around 95% of our human isolates belong to this subgroup. And to give you an idea of the most common serotypes that were identified in the cases with reptile or amphibian contact, Salmonella typhimurium was the most common, identified in 81 cases, and then followed by those listed in descending order on the slide here. I've asterisked the first 6 serotypes because, interestingly, each of these is a serotype for which mice are a reservoir, which is interesting because, as Alex mentioned, many reptiles do eat a diet that includes feeder rodents or potentially includes feeder rodents. I did want to note here, though, that because our analyses didn't adjust or control for other risk factors, such as foods or specific exposures that people might have had during the exposure window, association here does not imply causation. We were just interested in some preliminary investigative work. So looking more closely at the reptile types reported by cases of each serotype, there also appeared to be some associations between serotype and reptile type. So there were 16 human cases of Salmonella paratyphi biovariant Java in our data set. Almost 90% of these reported contact with a snake. All of the Salmonella Chester cases, most of those with Salmonella caudum and Salmonella Tennessee specifically reported a bearded dragon, and 4 of 5 Salmonella agbeni cases reported contact with a turtle. Looking at the literature, these associations do appear to be plausible, as there have been known outbreaks in Canada, the United States, or elsewhere, specifically associated with each of these reptile and serotype combinations. We then looked at the reptile isolate data for the same time period, and we were interested to see if we saw the same serotype and reptile associations that we did in the human data set. Here, there were 35 unique serotypes, with most of these, again, only reported once. Similar to the human isolates, most were also Salmonella enterica subspecies enterica. However, Salmonella enterica subspecies arizonae was also common. This was not unusual because reptiles, and particularly snakes, are a natural reservoir of Salmonella arizonae. So it can, however, cause gastroenteritis if transmitted from reptiles to people. And in our human data, there were a small number of case isolates belonging to this subgroup. I'll pass it to Alex quickly just to talk about the reptile isolates.
So then we say reptile and amphibian veterinary isolates from Ontario. So we looked at the same time period. It was 2015 to 2022. We didn't actually have any amphibians tested at this time. They tend not to make it to veterinary care, and they tend not to be submitted for post-mortem. We did have 46 salmonella isolates from reptile specimens reported to us in the time period. 80% of them were privately owned reptiles, salmonella being most commonly isolated from bearded dragons, again, reflecting their popularity as a pet, particularly in households with children. And about 20% of our isolates were represented by zoological institutions, most commonly coming from Eastern Massasauga rattlesnakes because there is a recovery program for them. A number of them undergo surgery, get implants. They're generally handled quite a bit, so it's not really surprising that when there's any illness, they were tested. Most of the salmonella isolates come from snake submissions, and again, as things that eat obligate diets of whole prey might reflect a rodent diet as well. Next slide, please. So the obligate carnivores were most likely to have salmonella isolated. We had 32, so that was about 70% compared to our omnivores, so things that ate both meat and vegetable matter, or things that were strictly herbivores, which were the sort of least commonly represented at 9%. And we did have some that weren't really identified at a species level, so we couldn't figure out what they are. As I said, all snakes are obligate carnivores. Mice are also a reservoir for salmonella, so are rats. And this probably reflects that their diet makes them at high risk. Bearded dragons are very commonly bred overseas, in tropical countries where they can do this outdoor year round, not have to invest in heating. And then they're imported to Canada en masse, and then they're widely distributed through retail pet chains. So again, this is a significant stress event. They're being raised in low biosecurity, and then sort of wide distribution across Canada might reflect that they're sort of higher risk for causing human disease. And then this increasing interest in the powder to gel diets, the alternative insect species, artificial prey, this can also drive salmonella infection.
All right, so based on all of this information, what are our takeaways for public health folks? So we'd recommend using the standardized questionnaire for Salmonella to guide case interview. If a case does report contact with reptiles or amphibians, we'd recommend attempting to gather all relevant information. This can be really helpful for risk assessment and kind of giving an indication if it might plausibly have been the source of exposure. If you do suspect reptiles as the source of illness in a case or outbreak and are interested in exploring this further, our lab folks are available to help, so please reach out before you collect specimens If you do plan to conduct testing, we would suggest environmental swabs just due to the limitations associated with cloacal swabs and fecal specimens and intermittent shedding. And if you are a reptile owner or considering getting a reptile, not to put you off having one of these as a pet, but just to provide some considerations to be aware of. So just recognizing that they and their feed are a potential source of salmonella. So the importance of hand washing, environmental cleaning and disinfection, supervising interactions with children and reptiles, particularly if you do have a small turtle and you don't want your child putting it in their mouth, and ideally restricting roaming and having dedicated containers and feeding implements. So we'd rather see somebody bathing their bearded dragon in a reusable dedicated container like this, rather than in your kitchen sink or bathroom sink where we'd have to worry about cross-contamination. And lastly, if you're a reptile breeder or seller providing advice to someone who is, similar considerations, but also prioritizing preventative veterinary care and making sure that you're providing infection control information to clients at point of sale, just so that they have the information that they need to take appropriate measures to prevent infection for themselves and others in their household. What we find a lot is that folks, as Alex mentioned, don't recognize that the reptile feed is a potential source of exposure or that indirect transmission is possible. So for example, not recognizing that if you're holding an infant and we're just holding your bearded dragon, that you really should be changing your clothes as well as washing your hands in between. Otherwise, your clothing can serve as an indirect source of exposure. So I will skip past this, but just summarizing basically everything we've reiterated. We want to make sure we have time for questions, have provided references if you're interested in reading more, and just wanted to take the time to acknowledge our co-authors who are listed here just because we did luckily have a successful publication, which we are excited about. If you have any questions, please feel free to reach out via the email address on the screen. Otherwise, I'm going to hand it back to Christine to facilitate our Q&A. Thank you very much for your attention.
Thank you, Dr. Paphitis and Dr. Reid, for the insightful presentation. We'll now move to the Q&A segment to answer questions from our live audience. And a quick reminder to everyone, if you have not already, please enter your questions into the Q&A pod. So while people are getting their questions ready and entering into the chat pod, I do have one or two questions that I'd like to ask of the speakers. Okay, so we learned today about how either the reptile or the dietary sources might cause salmonella infections in humans. Can you tell us a bit more about an outbreak that involved pet reptiles and how it was determined what the source of the outbreak was, the pet or the food? And did the predominant isolate give us any clues in that investigation?
That is a great question. We've had several, so maybe I'll start, Alex. So this is actually what piqued our interest. So we've actually gone down a bit of a rabbit hole with reptile-associated salmonellas, and we've got a little series that we've been writing about. There was one outbreak that we had with bearded dragons, I believe a year or two ago now, where we noted that there was a common risk factor mentioned in that all the implicated cases reported having contact with bearded dragons in their household. So not to pick on the poor bearded dragons, we also did a summary and looked at other potential commonalities, including food exposures, common like meals that they might have shared and things like that. And it's important to keep an open mind and make sure that you're not focusing too narrowly on one potential exposure right at the start. So we do recommend collecting leftover food samples, but in this case, we took environmental swabs. We did try fecal specimens. This is one of the earlier ones that we did. And we have tested feed before. So for example, if someone feeds crickets, we can swab the container that the crickets were in and test that as well. And in this particular outbreak and in others, it's the environmental swabs that we found to be the most reliable. Just, I guess, the temperature and because of the type of surface material, it does facilitate detection of salmonella longer than if we were collecting a fecal specimen, just because of that intermittent shedding issue. So yes, we don't only test these things, just wanted to make sure clarifying that we do also test food, and we will look at all potential exposures in our risk assessment. And then if health units choose to pursue the testing of the reptiles, or we would encourage that if it is looking based on the epidemiology that might be a potential source, then that can be very helpful. I don't know if Alex wants to add anything. And then whole genome sequencing obviously is super helpful, particularly if we can isolate the same serotype and the same whole genome sequencing pattern from the human isolates and from the reptile isolate.
Yeah, thanks, Katherine. It really highlights the need for that epidemiological information the environmental investigation as well as the laboratory component, and how those things all kind of get tied together to help solve the outbreak. I have a couple of other questions from the audience now. I have one here that says, do you have any recommendations for managing pet visits for healthcare facilities that may have residents or patients that request pet visits with reptiles?
That is a
can
great question. Go ahead, Alex.
I can start. So like any other animal, it's best to work with someone who has routine health visits with a veterinarian, make sure the animal is healthy, doesn't have any other issues going on. And then, if there's any concern that, like, for example, these are patients who may not be fully mobile, making sure that you are somehow able to disinfect their hands, not have it, contact things like wounds, or, that there's some sort of supervision going on, bringing them in a clean enclosure, say on a clean towel, that kind of thing, just to minimize how much salmonella you might be bringing from the enclosure with the animal.
Yeah, thanks, Alexandra. And I know it's been like over the last number of years, it's also been very popular to bring in these types of animals to like say daycare centers where we know that the risk of becoming infected or having more severe illness is often higher for these
Yeah,
children.
I would not allow small children as much as possible to handle, especially if you have like 30 children, right? It should be more of a show and tell than
Mhm.
having every child handle those animals, just because you can't keep on top of 30 kids, making sure no one's putting their fingers in their mouths, that kind of thing, right? It's hard enough to do hand hygiene with small children.
Can I
That,
just add, if
go
someone
ahead.
is in a daycare or you're considering having like a birthday party with reptiles, my kids have been to those, and I was that parent running around with the hand sanitizer, making sure hands didn't go in mouths. What we'd suggest is if you're in a facility like a long-term care or an acute care setting or a daycare, that you reach out to your public health unit, ideally before bringing any animals in, just so you can have a conversation about risks basically, and if there's some animals that are not recommended for certain populations. So for childcare, for example, reptiles would not be recommended for children under the age of 5. There's safer alternatives, but if you are going to do something like this, having the considerations that Alex mentioned, hand hygiene, making sure the animal's healthy, having like a changeable barrier between the person and their clothing, so a towel or something else launderable or disposable, and then ideally not having it travel around the facility, doing it in a central location, and then ideally outside. So if it's a birthday party, take it outside and do it on the grass instead of having inside where you have to worry about contamination of surfaces within the household. So things like that.
Okay, and here's another question from the audience. What can reptile owners do to seek out feeder rodents or insects raised according to best management practices?
It's tricky. I mean, they're going to have to ask questions of the person who supplies them. So for insects, there's not a lot you can do. These are generally raised sort of in specialty environments. Like there's big cricket factories, but there are only a handful of them. They tend to come through central distribution systems to pet stores, for example. So I think just recognizing that it can be a risk and managing that risk and how to sort of do proper food handling at home with them in their feeder rodent side. But if you have more animals and you're buying directly from someone, like I buy my chicks from a guy in Cambridge, or I know who supplies my rodents, ask questions, right? Like you can ask to see photos of how they raise their animals, make sure that they're sort of more aligned with best management practices than some of them that are kind of terrifying if you go inside them.
Thanks. And I
I don't
think
know if,
it, oh, sorry, go ahead,
I was going to
Katherine.
say if Catherine
Oh,
has anything.
yeah, and I think the other point that you highlighted earlier, Alexandra, is the complexity of their dietary needs that may not be met with just one single dietary source. So it does sound very challenging to make sure that you're covering off all of those bases in terms of dietary needs, like best management practices, as well as trying to minimize the risk to like the humans in the environment.
We're trying to work on some sort of fact sheets for reptile pet owners through our Ontario Animal Health Networks. I just haven't gotten to them with summer and covering vacation or being on vacation, but they are coming at some point, hopefully this fall.
Yes, I did wonder if you had a good sense for pet owners' knowledge about the risks associated with pet ownership and enteric disease?
I think it's pretty low, honestly. Like we struggle with it a lot across multiple species, right? Like dogs and cats on raw food diets are one of our nightmares, right? There's a lot of misinformation on Facebook or wherever people are getting their information from. I think a lot of people, because like I know people know that they should cook their chicken well, but generally people aren't as worried about the food they buy for themselves. So they think all these things are safe, right? Because we've got a robust food safety system on the human side. So why isn't pet food the same? And I don't know if they realize how unregulated these industries are and that there's a lot of sort of buyer beware out there.
I think the raw pet food diet it would be another great follow-up presentation for you two to do, because I think there's a lot of misinformation, and we have seen some of this evidence related to outbreaks associated with this particular dietary type, and I think some of it is related to sort of a lack of understanding among pet owners, and maybe some in addition, possibly vendors as well, about the potential risks to the pet owners. I have a question from the audience here. For human case isolates, was reptile and amphibian contact specific to those who handled the reptile or amphibian themselves, or does living in a household with a reptile or amphibian also count?
Great question. So typically for the routine case investigation, our standardized questionnaire that most investigators use, or they have a local equivalent, asks about contact in the previous 7 days. And so usually we ask about in the household, like, do you have one of these as a pet? And then depending on the investigator, usually there's a little bit of back and forth with the client about just transmission and that it is possible for direct or indirect. In the event of an outbreak where we have a hypothesis, or we think that, for example, we might have a Salmonella outbreak that's reptile associated, ourselves or the relevant province or territory or our federal counterparts often will re-interview and ask more probing questions. So a lot more detailed questions about direct or indirect contact, types of food, and things like that. So the preliminary questionnaire just asks about basically the presence of a reptile or having any type of contact. And then if we do more in-depth follow-up, then we get more into the nuances. But yes, we have seen in our previous outbreaks both a skewing towards younger age groups, like a disproportionate number of cases being younger, particularly under fives, tend to take up a large proportion of our case counts. And then once we ask those probing questions through re-interviewing, there does end up being a fair number of cases, or a decent proportion, that do only report indirect contact. And we have had outbreaks where the cases are occurring in those that we know have had indirect exposure. So for example, through caregiver clothing or contact with environmental surfaces where a reptile has been allowed to free roam, just because there's that lack of awareness of how it's actually transmitted, or that they have Salmonella potentially present in the first place, even if there are no clinical signs.
Yeah, thanks, Katherine. I think I recall a scenario where, say, let's say the teenager, it's their pet, they're responsible for the feeding and care of the pet, but they've been washing like the food bowls in the same sink as where everybody else is in the kitchen, as opposed to, say, a utility tub or some other dedicated space for that animal. So even if everybody else is not coming into contact with that animal, and somebody else is completely responsible for it, other people are unfortunately getting that cross-contamination in the environment. So I think it's time for us to wrap up today. And I would like to thank Dr. Katherine Paphitis and Dr. Alexandra Reid for presenting. I'd also like to thank everybody who joined us for today's event, and especially to those who asked questions. You can expect to receive a brief and anonymous survey for today's session. Please try to complete this to help us improve our programming. Thank you, everybody, and have a wonderful day.